Hello everybody and welcome to the last lecture of today's course here. So now at the end of the day, I hope you will still stay awake another hour, stay with me, because now the topic of the last hour of the day will be all on PTMs, on post-translational modifications, and on phosphosite localization using targeted proteomics. So I have structured this presentation into two parts. In the first part, I would like to again give you a rather broad introduction into what PTMs actually are and why they are interesting. I would like to discuss with you the typical workflow that we apply in terms of sample preparation and spec measurements when we do PTM analysis. And I would like to end this first part with some key technical challenges that we are facing currently in PTM studies. And that will also then bring us directly to the second part, where I would like to look with you into one specific challenge, which is the localization of PTM sites. And we will look at one real case study from myself where the purpose was to position phospho sites on peptides. And in the very last part, I would like to just briefly show you a little bit how PTMs can be set up in Skyline itself. Okay, so let's get started with part number one. Whenever there are questions arising from your side, please don't hesitate and just interrupt me at any time. So what is a post-translational modification? I guess in this audience here, I do not really have to introduce that, but let me just very briefly summarize. So after mRNA gets translated by the ribosomes, the formed polypeptide chains are often not the ready mature protein, but they need to undergo further processing before they become the functional mature protein. And these further processings is what we call post-translational modifications. And what most people consider or think about when they speak about a post-translational modification are those modifications that are reversible additions to chemical groups to either the N terminus or the C terminus or to the side chains of proteins. And so the typical examples of this reversible addition of chemical groups are, for example, phosphorylation, methylation, acetylation, or oxidation. But next to these reversible additions of small chemical groups, there are also a variety of other PTMs that are occurring on proteins. For example, the reversible addition of another protein, another polypeptide chain to a protein. Typical examples here are, for example, ubiquitination or simulation. But the cell can not only attach large polypeptides at a PTM, but cells can also attach other large complex molecules in a reversible way, like, for example, sugars or fatty acids, or the cells can do ADP ribosylation as a PTM, a reversible PTM. And while these three examples here that I name are all typically reversible, so the cell can attach or remove these PTMs from polypeptides, there are also irreversible PTMs ongoing, for example, cleavage, irreversible cleavage of the polypeptide chain by proteolysis, so by proteases. They can cleave off the N, very often N or C terminal signal peptide sequences of proteins, or another less well-known irreversible cleavage of proteins is called intein splicing. And a lot of people know the splicing ongoing on mRNA level, where introns and exons are working, and where introns are spliced together and exons are spliced out. And something very similar can actually also take place on protein level, where we have intein and extein sequences, and exteins are spliced out and inteins are spliced together. And that's also an irreversible PTM that can happen. And then finally, we have also irreversible amino acid modification. So deamidation, for example, is an example for this type of PTM. So what this whole slide, or this whole this whole what this whole slide should convey you is that there is a huge variety of very different PTMs that are chemically, the chemical structure very, very different, and that yes, this huge variety of PTMs really leads to proteome diversification. So cell life uses this huge variety to generate very different functions and very different chemical structures on proteins. Now, why are these PTMs interesting and what do they influence? So almost every protein gets modified at least once on one or even several positions during its lifetime. And this whole process of attaching and removing PTMs on one or several sites in a certain time space is what we call the PTM code. And this PTM code really regulates and fine-tunes the function of proteins in time and space. And actually, I think all the protein functions that you can think of a protein can fulfill have been already been associated to a PTM function. So here's just a starting list of functions that are influenced by PTM: cellular localization, activation or deactivation of enzyme activities, signal transmission, cell-cell interactions. PTMs can change the conformational state of a protein, its solubility, its stability. PTMs are involved in degradation and many, many more functions. So PTMs are involved in pretty much everything you can think of. And unraveling the PTM code, so understanding when a protein gets modified in time and in space, and how different PTMs are actually communicating with each other, how they crosstalk with each other, is one of the great challenges that molecular biology faces at the moment. And the fact that this is a currently very, very interesting topic, you can also see from this slide here, where we plotted the number of PubMed publications that contain the term PTM, either in the title or in the abstract, by year, and you can see that there is an almost exponential increase in papers that describe and publish PTM work. So it's really a very, very state-of-the-art important topic to study. Now, how can PTMs and proteoforms, so a proteome form is, what I mean with a proteome form is a protein polypeptide chain, the same polypeptide chain that carries different PTMs. So how can they be analyzed? And there are two major analytical strategies that people use to study PTMs. The first strategy is to work with antibodies, with PTM-specific antibodies, and to then do Western blots or Luminex assays or protein arrays to study these PTMs. So there are antibodies that are either specific towards a general PTM, so there are antibodies to enrich for acylated lysines, for example, but then there are also antibodies available there that are specific for certain PTM sitting on a specific peptide or a specific protein. And all these antibody-based methods can be very valuable and can work very well. You can measure antibodies very sensitive with Western blots or with this type of assays, and you can also multiplex greatly. But the big disadvantage of these antibody-based methods is that you need a well-working antibody. And to have well-working antibodies for PTMs is even more challenging typically than just having antibodies for specific proteins. And that's why definitely the Rolls-Royce King method, analytical method when it comes to PTM analysis is mass spectrometry. And the reason why we can use mass spectrometry to study PTMs and peptides is because every PTM changes the mass of the peptide of the protein. You always add or remove a certain mass due to a PTM, and by specifically measuring this mass shift that we can see here, a small list of example mass shifts. By measuring these mass shifts, we can identify and quantify PTMs with mass spectrometry. Now, Here you see the typical workflow that people apply for bottom-up proteomics. And this workflow, of course, looks very familiar to the workflows we already discussed during the day today and yesterday. It's the typical bottom-up proteomic workflow that people also apply for normal proteomics or analyzing naked peptides. But there are in each of these steps some specific considerations that you can think about if you want to do a PTM analysis. And on the next slides, I would now like to give you some specific thoughts or tips, tricks that you can think about in each of these steps when it comes to PTM studies. So let's get started with the first step of protein extraction. So when you start working with some type of sample, it is always, of course, super important that you efficiently and reproducibly extract ideally all proteins from a sample. However, when it comes to PTM analysis, this extraction step is especially important because there is one very important thing to consider here, and that is the fact that the PTM status of a protein can change very rapidly during this protein extraction step compared to when you do normal proteomics, where you want to determine just the concentration of a protein. It is actually, I think, fair to say that the total concentration of a protein in the cell does not change that rapidly. So if a certain protein should be up or down regulated by the cell due to a certain stress, you always need a certain amount of time, several minutes, maybe an hour, until the whole translation and transcription machinery has started. So protein concentrations are fairly constant over a certain time frame, while PTM status of a protein can change really within seconds. So and this is the case because there is usually always an interplay of different enzymes in different proteins in cells that either attaches or removes PTMs from proteins. So in the case of phosphorylation, we have the interplay of kinases and phosphatases that attach or remove phosphate groups, or we have the interplay of acetyltransferases and deacetylases that attach and remove acetyl groups. And it is actually really true that these processes here can take place very, very fast. So an important point to think about when you do protein extraction for PTM analysis is that you must do this protein extraction under a condition where the status of your PTM is really maintained. And here you see just some examples of buffers or conditions that people tend to work with or like to work with in PTM analysis. So a standard extraction buffer also people work with is 8 molar urea, because this buffer denatures all proteins and thereby hopefully kills all protein activity. Or 6 molar guanidinium chloride is the same, or sodium deoxycholate. One specific thing that people often do when they work with bacteria or yeast, and when they want to do PTM analysis, is they add trichloroacetic acid to their growing cells, because TCA does also kill proteins, that it denatures all proteins and thereby kills all enzyme activity, which can also freeze and maintain the PTM status. However, you cannot apply such a TCA sampling protocol, for example, to human cells, because TCA would actually lyse the cells immediately, and then you could no longer centrifuge and wash them, something like this. But with bacteria or yeast that have this rigid cell wall, TCA sampling is something worth considering. 3,4-ethanol is another very famous or more and more frequently used buffer component, which was published in this EasyPhos protocol from the Matthias Mann group. You can also consider using PTM inhibitors, so kinase inhibitors or phosphatase inhibitors, or whatever is available for your PTM of interest. And then there is the strategy to try to work as cool as possible for PTM analysis. or just the opposite, heat your cell in your cells or your sample in order to inactivate all enzymes and to maintain the PTM status. So these are all suggestions and ideas and examples of what people do. And the point I want to make here is just that, yeah, when you plan a PTM study, this protein extraction step is something you should really think about, and you should maybe invest on optimizing and finding a good protocol for your sample and your PTM. The next step in every bottom-up proteomic workflow is an optional step here. It's the protein enrichment step. And this is something that you can do in order to improve the sensitivity of your measurement. So one big problem that we have when it comes to PTM analysis is that peptides that carry PTMs tend to be of lower concentration, because very often a PTM does not occur on a protein to 100%, maybe only to 1%. So that's one reason why PTM peptides tend to be lower concentrated than naked peptides. And then there is also the second problem, that peptides that carry a PTM might not be as good detectable in the mass spectrometer than naked peptides. This is, of course, something that is very PTM dependent, but it has been shown that, yeah, for some PTMs, just the ionization efficiency is not as good. And that's another reason why for PTM carrying peptides, we often get lower signals in our mass spec. And this is the reason why we need to improve on our sensitivity in our sample preparation. And one way to do that is to do protein enrichment. And protein enrichment protocols people work with is, for example, co-immunoprecipitation or affinity enrichments of certain proteins of interest. There is a well-established protocol for histone purification to specifically enrich histones, or you can work with different products that enrich specific proteomes, subfractions of your proteomes. For example, there are beads available that specifically bind kinases to enrich for kinases and so on. Okay, the next step in the workflow is again an essential step in bottom-up proteomics, is the proteolytic digestion. We heard already from Andy how important this step is, and how variable it can be, and how long you digest with trypsin, or whether you shake or not. From the perspective of PTM analysis, this digestion step is also especially important or interesting, because when it comes to PTM analysis, the one important thing you need to keep in mind is that you really will have to work on peptide level, or in other words, you don't have the freedom to combine or choose different peptides of a protein, and to combine information from various peptides to one protein information. No, in PTM analysis, you're really bound to peptide level. And it might well be that when you work with trypsin, that a certain peptide sequence is just not, or a certain patch of a protein is just not accessible for you. So I've brought you here one example. So let's assume you're interested in that tyrosine here, and you hypothesize that it's phosphorylated. So if you work with trypsin, which will cut after this arginine and this lysine, this tyrosine would actually sit in a peptide that is only a size of 5 amino acids. So it's very small, very likely unaccessible, not detectable by standard proteomics methods. So that would be bad. However, if you work with other proteases like Lys-C, you would generate such a peptide here with Glu-C, an even larger peptide, Arc-C, Asp-N. So you can try to find alternative peptides in order to increase the sequence coverage of your protein, and to find maybe better suited peptides for specific sites where you are interested in, or where you expect PTMs to occur. Okay, the next two steps here in the workflow are again optional, and they are both there in order to improve sensitivity of your measurement, just like protein enrichment. So one thing you can consider is to do a PTM peptide enrichment strategy. There are various different strategies out there for different PTMs. These three here are what I think currently the most frequently used PTM peptide enrichment strategies. They're all well established and well described. So, for example, there is an antibody available that recognizes lysine GG motifs, and this lysine GG motif, that's something that occurs when a peptide is modified with ubiquitin. And if you digest a ubiquitin modified protein, this GG sequence here that comes from ubiquitin will remain linked to the side chain of a lysine, and there are antibodies that recognize exactly this lysine GG attachment to the side chain very specifically. And like this, you can enrich for ubiquitin lysine ubiquitinated peptides. Another standard PTM enrichment protocol is the enrichment of phosphorylation using either titanium dioxide or iron IMAC type of strategies. Or another good example is acetylation, enrichment of acetylated peptides, acetylysine peptides, by again using an antibody that specifically recognizes these acetylysine groups. And these peptide enrichment strategies can be very powerful and then can really be a game changer for you, because you can achieve with a well-working protocol a really great enrichment ability to detect these peptides, but unfortunately not for every PTM type enrichment strategies exist. And the same motivation, improving your sensitivity for certain PTMs, is this optional peptide fractionation step. So it might be that you would like, in order to dig deeper into your PTM proteome, you want to fractionate your samples, and here people typically use exactly the same fractionation methods than people use also for naked peptides, like basic reverse phase fractionation, strong cation, strong anion exchange, stuff like that. Okay, the second last step of the workflow is mass spectrometry, and I think it's very fair to say that currently still most of the PTM work that is published is all data where the data is all projects where the data was acquired by data dependent acquisition, so by DDA. However, there are also alternatives to doing the standard DDA shotgun type of data acquisition. And these interesting alternatives are, for example, to use DIA as your method, data acquisition method, or to perform TMT labeling with your samples and then do DDA, MS3, and synchronous precursor selection type of measurements on the fusion Lumos instruments. Or, and that's why you are here and that we will talk about for the rest of the presentation, is to use targeted proteomics techniques, so like SRM or PRM, and use these methods to acquire the data. Okay, and the last step then is data analysis. And this last step I would like to use to state what I think are currently the major analytical challenges that we are facing when we look at PTM data that was acquired by DDA, so by shotgun proteomics. So one major challenge that we are facing is the issue of missing values and limited reproducibility. That's an issue that we already discussed in the beginning of the course, which is also equally valid for normal proteomics, or when you measure naked peptides. With PTM data, I think it's even more severe, because when you generate with DDA large data sets where you have a lot of samples measured, then with PTM analysis, we do not have the freedom to merge several peptides into one protein value, but the quantitative data results will remain on peptide level, and we will therefore just have a lot of missing values, limited reproducibility in these data sets. And then another analytical challenge is definitely sensitivity when it comes to PTMs. I said this already many times. PTMs tend to be of low concentration, and they tend to be, depending on the PTM, bad flyers in the mass spec. And that's all the reasons why the signals of the PTM carrying peptides tend to be low. And we need to be able to really be sensitive in our measurements to nicely detect these peptides. And then there is the issue of PTM site localization. This is an issue that might be very easy to solve for certain PTMs. So sometimes it is very clear based on a peptide sequence where a PTM happens, because there is just one amino acid that can actually carry this PTM. But sometimes it can be very difficult to localize a PTM. And the prime example here is phosphorylation. So phosphorylation, you all know, can typically happen on serine, threonine, or tyrosine residues, and almost every tryptic peptide, or the majority of tryptic peptides in a protein, contains several serine, threonines, or tyrosines. And then it can be very challenging to find the correct amino acid that actually carries this. And honestly, I also have to say that I think that this issue of PTM site localization here contributes to the issue number one of missing values and limited reproducibility. Because if you do a DDA analysis, and if the algorithm that you're working with is not capable of reproducibly putting a certain PTM to always the same amino acid in a peptide, but if instead, in sample 1, the algorithm says a certain PTM sits on serine number 3, and then in sample number 2, it puts the same PTM on serine number 4, so another serine, what will happen is that the data matrix that you will generate in the end will contain more phosphopeptides on the y-axis, right? You have more lines in there, but you will also have more missing values, because sometimes the algorithm puts the phosphopeptide describes or quantifies one phosphopeptide version, and in the next sample the alternative phosphopeptide version. So just as an additional comment, why this PTM site localization issue actually negatively affects also this issue of missing values. Yes, and then a last challenge that we have in PTM is the challenge of studying different proteoforms, and because as long as we actually apply this workflow that I show you up here, this bottom-up workflow where we digest our proteins with peptides, we completely lose with this workflow the information of how peptides occurring, of how PTMs occurring on different peptides are talking, or we are talking to each other. Because at the moment we do bottom-up proteomics and we digest, we do not know where certain PTMs were sitting on different proteoforms. But if we really want to unravel a PTM code, and if we want to know how PTMs are occurring on different positions of the proteome, of a protein, then we would need to stop digesting our proteins and start doing top-down proteomics to really measure complete proteoforms. And that's, as we all know, very, very challenging. Okay, so what I would like to now show you next is I would like to show you how targeted proteomic methods such as SRM, PRM, or DIA can actually help to improve on these first 3 analytical challenges that we face with DDA. And I think we already discussed a lot how these methods can improve the issue of missing values and reproducibility and sensitivity. And therefore, I would from now on like to focus on this PTM site localization issue and how we can improve on better localizing our PTM sites with targeted proteomics. Okay, so this brings me to this part 2. And I would like to start this part 2 with a real life case study from my own research. And hopefully this will help you in understanding or give you a feeling of this problem and how this problem of site localization can be tackled. So one of the projects that I worked on during my postdoc was to investigate phosphorylation changes in yeast upon osmotic shock. So what I did at that time was I grew yeast in cell culture in various replicates in a normal medium, and then I added salt, high concentration of salt to the medium, and measured, grew the cells in that salt medium. And the purpose was to compare the phosphoproteomes between these two growth conditions. So what I did at that time was to perform the standard phosphoproteomic workflow that we had established in the lab. So I sampled my yeast cells here with TCA, with the trichloroacetic acid protocol, and with 8 molar urea in my extraction buffer. I digested with trypsin at the time, enriched the phosphopeptides with titanium dioxide, and then I measured those samples in shotgun proteomics data-dependent acquisition mode on NLTQ Orbitrap, and as a result, I generated then this quantitative results table where I had different phosphopeptides here in my lines, and I had different quantification values before and after the osmotic shock. So, and what you see here on this slide is some sections from this original quantitative results table that I generated at that day. For 2 different peptide sequences, one coming from the protein GPD2 and the other one coming from the protein GPD1. And what was special about these 2 different peptide sequences was that they occurred multiple times in my results table in various different isoforms. So for example, this peptide sequence here of GPD2 contained 3 possibly phosphorylatable amino acids, namely 3 serine residues, and I found all 3 different versions of single phosphorylated peptide, of this specific peptide in my sample. Plus, I found 2 doubly phosphorylated versions also identified and quantified. And for GPD1, the situation was even more complicated. So this peptide sequence here had 5 serine residues, with 4 of them being in a row directly next to each other, and then the 5th one, and 4 different versions, singly phosphorylated versions I could identify and quantify, plus 1 doubly phosphorylated form. And when you look here at the quantitative information that I got from this shotgun data, you can see that I got some missing values for these different phosphopeptides, typical problem. But when I averaged the intensities over the replicates and looked at the ratio, I could find that actually some of these phosphopeptides seem to be downregulated between the 2 conditions, while others seem to be upregulated, while again others seem to be not regulated at all according to the cutoffs or the thresholds that I defined. And when I saw this data at the time, I immediately asked myself, are really all these identified phosphosites truly occurring in my sample? Does the cell make all these different phosphopeptides? And do they then also really show different regulation? And does this really reflect the true biology that goes on in these different cells? And one thing that I could have done at the time, which I didn't, in order to maybe bring a little bit more light on whether these phosphosites here are present or not, would have been to do a site localization, to estimate the localization probability. And there are various tools out there that can do that on DDA data, like for example Lucifer or A-score or Tormeda or PhosphorS. So these tools, they can calculate for you based on the data how likely a certain phosphopeptide is really localized there. But what we wanted to do instead at the time was to use targeted proteomics to validate these shotgun results. So what I did was I went shopping for phosphopeptides, for crude, heavy labeled phosphopeptides, which are just in the range of 20 to 40 euros per peptide, so not very expensive because they are not purified. They are still crude, but they are heavy labeled at their C-terminal lysine residues. And I shopped all the 3 different versions of the singly phosphorylated GPD2 peptide, and I shopped all the 5 different singly phosphorylated versions of GPD1. And once I had these synthetic phosphopeptides in my hand, I then wanted to start doing an SRM experiment with them. And therefore, I looked at the transition list for this peptide and I realized, wow, shit, a lot of these Y and BI fragments, which you see here in this table for the 3 different phosphopeptides, are actually shared, are exactly the same. Namely, all the transitions shown in red are shared, and only the transitions in green here are transitions that are unique for one form or for the other form. So the precursor M over Z of all these 3 peptides is of course exactly the same, because they all have exactly the same chemical formula, right? But the difference of these peptides really just occurs for specific Y and B ions where these different peptides are different from each other. And when you look at this table, you can see that there are 2 unique Y ions for this first peptide, serine 70. There are 3 unique Y ions for this 75, serine 75, and there's not a single unique fragment ion for serine 72. So now you might think, oh, this is really bad, right? So there's not a single unique transition for serine 72, so I will never ever be able to say for sure whether this peptide is present or not. And this is true if we would be able to only measure, or if you would measure with SRM only a single transition per peptide, and there would not be a single transition that would be unique for serine 72. However, what we do in SRM is we always measure several transitions, and what you can do is to use a combination of transitions that is unique for this peptide, and that's, for example, the combination of Y9 and Y8 and B2 and B23. This combination is unique for serine 72. It does not occur in serine 70 or serine 75. So, and that's what we call, and what I will call in the rest of the lecture, a unique ion signature. Unique ion signatures are combinations of fragment ions that enable the correct PTM site localization. And depending on the sequence, depending on how many serines are there and how close they are, or how many other phosphorylatable amino acids are there, and how close they are to a certain sequence, unique ion signature can be only one transition, or it can be several. Okay? So what I did then was I measured these 3 different phosphopeptides of GPD2 by SRM, and I started doing that by measuring them all individually. So I used 3 different samples, and I just measured sample 1 with only this phosphopeptide present, sample 2 with just this phosphopeptide, and sample 3 with this phosphopeptide. And what I learned from this measurement was that every phosphopeptide here had a quite distinct and unique fragmentation pattern. So you can see that the colors here of the different fragment ions are really unique, quite different between these different forms. And what I did then was I made a mix of these 3 phosphopeptides. I put them all together into one pool, and I measured the pool in a 40-minute gradient up to 45% buffer B, and you can see that under these conditions, it could almost baseline separate these 3 different phosphopeptides from each other, right? So from this initial experiments, I learned that all these 3 phosphopeптide forms of this peptide are separable in retention time, and I learned that everybody shows a distinct, unique fragmentation pattern, and I learned how this fragmentation pattern looks like. Let's have a look at the more difficult example of GPD1. So also for GPD1, I had all the 5 different phosphopeptide versions measured individually. And you can see also they all showed a unique fragmentation pattern. And when I mixed those 5 peptides together into pools, I got quite a complicated mixture and overlay of peaks. However, what I learned there was also that actually I got something like 4 shouldered peaks, but the peptides were not, most of the peptides were not perfectly coeluting. They were still somewhat separable, even though far away from baseline separable. But there was one combination of perfect coelution, which was the peptides serine 24 and serine 25. These 2 peptides really gave almost a perfect coeluting peak. The others were somewhat separable. So also here, distinct isoforms, distinct fragmentation pattern was observable. but the peptides were not resolvable by retention time, as in the example of GPD2. They were much more overlapping, and serine 24 and 25 were not separable at all under the conditions that I applied. Okay, from this learning, I could already draw the conclusion that if I would use shotgun or DDA proteomics and do MS1-based quantification, it would actually be impossible for me to distinguish shotgun proteomics between these highly overlapping, and especially between these co-eluting serine 24 and 25. Because when you do shotgun proteomics, you perform quantification based only on the precursor mass, and the precursor mass is exactly the same for serine 24 and 25. So it would be impossible on the MS1 level to tell them apart. And what I would like to show you now is that when you work on MS2 level, like we do in SRMPM or BIA, what you can do is you can focus on fragment ions that are specific for the one and the other form, and thereby you can be potentially be it will potentially be possible to quantify also perfectly co-eluting peaks, to tell two perfectly co-eluting peaks apart from each other. So for our little example here, this would be, for example, the case for serine 25, serine 24, which were these two phosphopeptide versions that showed perfect coelution. We have a look at what are the differentiating fragment ions between these two different forms, and it's actually the Y5 ion that is different for these two forms. And when we just focus on this Y5 ion, and if we first look at Y5 541, that's the precursor that we would expect in these first three versions, then this is exactly what we see. The individual mixes, this transition is high in the first three isoforms, and in the mix we see this shoulder of three different peaks, while when we focus on the Y5 621, we can see this transition is only present in the last two phosphopeptide peaks, and you can see in the mix that these two phosphopeptide versions are actually quite nicely from each other, baseline separable. So now in this example, I could nicely quantify using this transition serine 25, even though it's perfectly coeluting with serine 24. Does it make sense? Okay, to once more repeat that and make this very clear. What I hope what you understand from this presentation is that in order to localize phosphosides and to tell phosphosides apart from each other, retention time information actually really matters. Because if you have a case like this here, a phosphopeptide version with three different phosphorylatable amino acids, then it really matters whether these three different versions are coeluting or whether they are separable in retention time. If they are separable in retention time, you might get a picture that looks like this. And now you can use this picture the unique transitions like this red one or this orange one to tell which of the three different groups is which phosphoside. And once you have identified which phosphopeptide is which group, you can use all of those transitions to quantify that peptide, right? So you have separation in retention time. You can use all fragments to detect and to quantify a peptide. Use the unique ion signatures to localize which of these peak groups is which site. However, in the moment when peaks are coeluting, when they are no longer separable in retention time, then the situation is different, because if you now want to do accurate quantification of just one form next to all the others, you can only use the unique ion signature signals to do that. So it's no longer possible to use all the transitions for quantification. You must be sure you use only the transitions that are not integrated. So the first lesson that I learned from working with a lot of these synthetic phosphopeptide was that accurate chromatographic retention time information really matters, and that it makes a difference whether they are coeluting or whether they're separate in retention time, which transitions you can actually use to quantify. And then there was a second lesson that I also learned from the study, and that regards neutral losses. So if you have ever done a bit of phospho analysis with shotgun proteomics and looked at a few MS fragment spectra of phosphopeptides, then you know that very often in this MS2 spectra of phosphopeptides, you get nice intense peaks that contain a phosphate loss, which means this is indicated here in scalar with these minus 98 masses. And what these phosphate loss peaks mean is that in these cases here, fragmentation of the peptide has not only occurred in the peptide backbone, but also the phosphate group at the side chain has been fragmented off. So this is demonstrated here. So what can happen with phosphopeptides if you fragment them is that this peptide, this bond here between this carbon and this oxygen actually breaks, and then you will generate a dehydroalanine amino acid at this position. Right? And when I saw this initially, I thought, wow, that's cool. I can use this neutral loss phosphopeptides because they are generating an unnatural dehydroalanine amino acid. So whenever I find a dehydroalanine amino acid in my data, I know it has been a phospho serine or serine here in this case before, and that means I can localize the phosphosite. However, when I worked a lot with these synthetic peptides, I realized that there is also an alternative reaction that can go on that also produces these dehydroalanines, and this is simply the water loss of a non-phosphorylated serine residue. So also in normal serine residues, this bond here can sometimes break, and then you also generate a dehydroalanine. This happens usually to a much lesser extent or less frequently than with phosphate groups, but it definitely occurs. It can definitely happen. Depends a lot on the sequence of the peptide, how frequently this happens. And it does occur. So to make this once more very clear based on one example, so let's assume you have this peptide here that is phosphorylated on that serine. You think it's phosphorylated on that serine. And if you would now use the Y7 fragment ion, where also this bond here broke, you will have the dehydroalanine at this position, and the mass of the fragment ion will be 785.51 MOZ. So if you see this mass, if you use this fragment ion or this transition in an ester MSA to verify or to validate the phosphorylation position, this would be very dangerous, because if you would have exactly the same peptide present phosphorylated at this serine here, the same fragment ion can also be generated by a water loss of that serine. You will end up with the same fragment ion. Both of these peptides have the same precursor. So it is actually not a fragment ion that you can use to localize the phosphosite. And that's also a lesson that I learned. If you want to determine phosphorylation sites, you should not work with this neutral loss fragment ions. You should work with the fragment ions that still contain the phosphate. Okay, and on this last slide, I just would like to show you now that actually all this work that we invested into ordering the synthetic phosphopeptides, measuring them first individually, and then mixed and looking at all these chromatograms is really worth the effort, because we used now exactly what I just showed you, what we learned, to quantify phosphopeptides in real samples. So what I did at that time was I used my phosphor-enriched yeast cell lysates, we were light, and I spiked in these mixes of synthetic peptides into the sample. And as they were isotopically labeled as acetomas, I could directly spike them in. And then I measured heavy and light peptides together. And that's how it looked like. So here you see the skyline chromatogram figure for GPD2. In blue, you see the 3 peaks for the 3 synthetic peptide regions that I just spiked in and that I just showed you before. And in red, you see now the light channel. So these are now the phosphopeptides coming from my yeast extract. And you can see very beautifully that I only got coelution and matching fragment ion intensities with 2 out of the 3 phosphopeptides, namely with serine 72 and serine 75. And we did this then in replicates before and after the osmotic shock, and we found a slight but significant downregulation of both of these phosphosites. And also for TP1, we could find a good solution, a good finding. Also in blue, the mix of the 5 heavy synthetic phosphopeptides, and in red, which was this perfect coelution of 2 and the strong overlap of 3 other versions. And in red, you see now the light phosphopeptides coming from the cell extract, and you can see again I have coelution only with 2 of the 6 by looking at the relative fragment intensities. I could unambitiously identify that I have only the phosphopeptide serine 24 and only the phosphopeptide serine 27 present in my sample. And as both of these endogenous samples here were now baseline separated, I could use all the transitions to quantify them. Exactly, and we found that these 2 phosphosites here were actually upregulated upon osmotic shock quite significantly, and we then did also in this publication here in MSB from 2012, already a long time ago. So there we then did some follow-up experiments where we mutated these phosphosites and could then also once more validate in these with yeast mutants the function and the validity of the importance of these specific phosphosites. Okay, so at the end of this presentation, now I would now just like to show you a little bit in some of the live demo how you actually make your Skyline document ready to understand and to work with details. And the important tab for you to go to is in the peptide settings, the modification tab, where here under edit list, you can actually select structural modifications. And you can see here from this, when you press this triangle here, you will get actually by default, Skyline will show you all the UniProt modifications that are available in UniProt, and our Phospho ST and our Phospho Y modifications are in there, so you can directly check them. And then Skyline will already show you the completely filled out edit structural modification tab. So for Phospho ST, you see that Skyline says this is a structural modification that can only occur on serine and threonines, not on any terminus. It should be a variable modification, which means that Skyline does not force it to be present on every serine and threonine. There can also be unmodified serine and threonines in your target data. And then here you see the chemical formula. So H3Os and 1P are the atoms that are added when serine or threonine gets phosphorylated, and it leads to monoisotopic mass increase of 79.996. And then there is this also very helpful and useful neutral loss button down here. So here you can specify specific neutral losses that occur with this type of modification. And Skyline will be able to understand and to work to show neutral losses. Exactly. And this type of, you can define like this in your Skyline documents any types of PTMs, either by selecting anything here from this drop-down menu, or you can also just generate your own PTM. If you're working with crazy glycosylation structures, for example, you can give it a name, define the amino acid where it should occur, and as long as you know the chemical formula that your PTM attaches to the protein, you can add them here, and then Skyline will be able to understand them. And as a last thing, I would like to show you, once you have set now this PTM, how do you get a PTM carrying peptide into your Skyline document, into the target window. And there are actually two ways how to do that. One option is that you simply insert via insert peptide or insert phosphate or whatever. You insert first only the naked peptide. And once you have a naked peptide in your target list, you can simply right mouse click onto that peptide and then select modify. And then Skyline will show you this window here where you get your whole peptide sequence. And you can choose here with this triangle here facing down, for every amino acid, the structural modifications that are defined in the settings. So you can say, okay, this threonine here I would like to have phosphorylated. You could also say this serine you would like to have phosphorylated. You can also say whether you would like to create an additional copy of this PTM carrying peptide and leave the naked in, or if you would not like to do that. And then Skyline will show you here in the target window by highlighting PTMs in bold letters and with this underscore here that it carries now a modification. So one way is to just get naked peptides in, right click on the peptide sequence, select modify, and add any type of modifications and combinations of modifications that you're interested in. This works very well. However, if you have a huge list of PTMs, PTM carrying peptides that you already know that's exactly what the ones you would like to work with, a faster way to get all of that into Skyline might be to use the function insert peptide. So go to edit, insert peptides, and then you do not just insert the naked peptide sequence, but you indicate already with squared brackets plus the mass that this modification that you would like to add adds to the peptide. So with phospho, this would be, for example, plus 80. So with these squared brackets and the plus 80, Skyline will understand that this serine here is the one that should be phosphorylated. And if you insert that now in your document, you will directly have the phosphopeptide at that position be inserted, and you can insert thousands and tens of thousands of phosphopeptides very fast, very specifically to Skyline. Okay, with this, I'm at the end of the presentation. Thank you very much for your attention, and I'm open to any more